How are you all? Good, wonderful. Me too. Week's half over already. These weeks are going fast. I think they're going fast. All right, it's great to see you all. I always like to start out with questions. Anybody have any? Anything you want more information about? All right, if not, let's begin. There it is. All right, let me know if you cannot see that. Terrific. All right, so I just want to remind you, we mentioned this before, and it's in the um, supplies list. But next week, we're going to do an activity together in class. So like I said, you'll see this in the supplies list, both the master supplies list and the one at the beginning of each unit. There's a supplies list for that week. So I'm not going to go over the whole thing today. I just want to, I know there's a lot of words on that page, but just to let you know that the activity that we're going to be doing. So that says October 14th. That's the wrong date. So it's next week, next Monday, or sorry, Wednesday that we'll be doing this. Now, as always, if you can't participate in this, this is not required. I just think it's a pretty cool activity. So if you can, it would be great if you could. What you will need are safety goggles, bromothymol blue, 3 small jars with lids or 3 test tubes with lids, a test tube rack if you're using test tubes, a drinking straw, a water plant, and water. So this bromothymol blue is a chemical. It is a safe chemical. It is an indicator that turns blue in the presence of oxygen and yellowish green in the presence of carbon dioxide. Here are some places that you can get it. The fastest, of course, is Amazon, but that's also the most expensive. So, there might not be time if you haven't looked ahead at the supplies list and ordered it, you might need to get it there. And then the water plant you can get at aquarium stores. I recommend going in and just telling them that you need a little tiny piece. You don't need a whole plant. You just need a branch of that plant. Sometimes they'll just give it to you. They'll just break off a piece and let you take it home. You might also be able to find an underwater plant if you have water near your home, a pond or a creek. It just has to be a plant that grows completely underwater in order to do that. All right, so we did this activity. We've been working on it in the regular lessons. And this is the one where you put the seeds in the bag with the hydrogen peroxide solution and then waited to see which one was going to germinate first. So remember what a controlled experiment is. What is that? What do I mean when I say a controlled experiment? Shmuel.
When only one variable is being changed and everything else stays the same.
Exactly. So is this a controlled experiment?
Yes.
Yes. We did our best anyway, right? It's hard to have a completely controlled experiment, but we did our best to keep the only variable that was being changed the quantity of hydrogen peroxide, the percentage of peroxide that we used. Now, as a class, this was not a controlled experiment because we did it in many different states, all your different homes. You had things that were different between them, so different kinds of seeds, different brands of peroxide. So as a class, we had a lot of variables, but individually, you had just one. So that would have been a controlled variable. So let's look at the experiment. Now, this was your second sort of unofficial lab report of the year. We broke this up into sections to make it a little bit easier to do, a little bit easier to understand. So reviewing the parts that you had to do, I want to talk about some of the common errors that I saw, both in this recent seed report and in the Jello enzyme lab. Now, if I were to say what the things that I saw that were the most common errors, I could narrow it down to one thing. You didn't follow the rubric. So when a teacher gives you a rubric, use the rubric. Look at what you need to do in order to get the highest point value, in order to do the best job you can, and try to get that. Try to do that. If it says use a numbered list, use a numbered list. If it says write your hypothesis, write your hypothesis. You get the idea. All right. Now, most labs are going to start with an observation and then some kind of scientific question or a lab question. That's the thing that gets us started. So we observe something and then we ask a question about it. So our lab question might have been something like, which concentration of peroxide will cause the seeds to germinate fastest? Now, the prediction comes next, and that's more informal. You don't always put that in a lab report. We don't always break it down that way. If your hypothesis is in if then format, it's going to naturally have the prediction in it. The prediction is what comes after the then. If this is done, then this is what I predict will happen. That's going to be in that way. So I think it's a good idea always to write your hypothesis in if-then format. So let's suppose that what we decided was one of these. We have our prediction and we have these for our potential hypotheses. All right, so this is just 3 that I picked from previous years. If seeds are put into a bag with the highest level of hydrogen peroxide, then the seeds will germinate faster. If the 5 milliliter bag works better for pepper seeds, then it is the best growing solution for seed germination. If there are higher concentrations of hydrogen peroxide present, then the seed will grow. Okay, I want to talk about some things that might be wrong with these and then some ways that we can make them even better. Remember that we're always doing in a hypothesis this kind of idea here. If something is done, then something will happen. All right, remember our friend here?
Independent variables are the ones you change.
That's all we need to hear. Independent variables are the ones you change. If you remember that sentence, independent variables are the ones you change, you already know what controlled variables are. All you need to remember then is dependent ones are what you measure. All right, now here's a couple examples here to review independent and dependent and what we mean. So independent variable is the one you change. In this experiment here, we are testing the way that light affects plant growth. What is our independent variable? What is the thing we're changing? Shmuel.
The amount of light that reaches the plants.
Exactly. The amount of sunlight is what we are going to change. So that's our independent variable. Then our dependent variable is going to be the growth of the plant. So a hypothesis that we might do, will you tell me, what would be a good hypothesis? If, then. If something, then something else. Umar.
If the plants get a moderate amount of sunlight, then they'll grow more.
Yeah, very good. Very good. Something like, if I give the plant more sunlight, then the plant will grow taller. Now, I could also do it the other way around. I could say, if I deprive the plant of sunlight, then the plant will die. Now, there's still something wrong with those hypotheses. What's wrong with those two? The use of the word I. We don't want to use first person when we do a lab report. This is a technical document. Most of the time when you do things for a class, your teachers are going to want you to use active voice. I did this, or I saw that. That's not true in a lab report. In a lab report, we want to use what's called passive voice. So instead of saying Paul used the Bunn method, we're going to say the Bunn method was used. Or, instead of saying I tested oxygen saturation, we would say oxygen saturation was tested. Now, the reason for that is because usually when we're doing a lab like this, the person doing the lab is not the most important part. You don't need to bring yourself into it. The most important part is the process. That's what we're trying to show here. So instead of saying, I ground or we ground this pig liver tissue, you say pig liver tissue was ground. Okay, that's your example. So to improve this, I could say, instead of if I give the plant more sunlight, then the plant will grow taller. If the plant is given more sunlight, then the plant will grow taller. Instead of saying if I deprive the plant, if the plant is deprived. All right. Now, I've been a little bit loose in accepting that active voice in lab reports. From now on, it's going to tighten up a little bit. You're going to do another lab report in a couple of weeks. So for that lab report, you will lose points if you use active voice. Again, not a lot because it's not the science that's involved here, but you will lose just a little bit. All right, so going back to these again, let's incorporate that if then, the independent and dependent variable. We're going to start with the independent. If I do something or if something is done. So let's look at the first one there. If seeds are moistened with the highest level of hydrogen peroxide. All right, how could we make that better? Throw out a possible improvement for that hypothesis. Well, what do I mean by highest level? Would someone read? I'm sorry, Lisa, go ahead.
Yeah, maybe like the highest level of concentration. Like you
Yeah.
could specify.
Yep, good. We could even get more specific and say what that level of concentration is. So I could say if seeds are moistened with a solution made with 30 milliliters of hydrogen peroxide and 250 milliliters of water. So that's even more specific. That tells me exactly how much I'm going to use, what it's going to be, rather than just saying highest level. Remember, the whole idea of all this is for someone else to be able to reproduce exactly the way that you did it. So would someone else know what you meant by highest level? No. Would they know what you meant by solution made with 30 milliliters of hydrogen peroxide and 250 milliliters of water? Yes, they would know that. All right, looking at the next one. If the 5 milliliter bag works better for pepper seeds, what's wrong with that? Or how could I improve it? Shmuel.
Wrong with that. It's not if something.
You're real hard to hear. Can you move closer?
Yeah. Also,
there you go.
It's my computer also. Yeah. It's the problem with it is that it's not if something is done, it's if something happens.
Right.
So
that's not telling me what I'm going to do, is it?
No.
Right. It's just saying, what do I mean by working better? You know, so that's not, I want to be specific. I want the if to be what I've done. So excellent. So I could say, if seeds are placed into the bag with 5 milliliters of hydrogen peroxide and 250 milliliters of water. Now that's doing something, but there's still something wrong with this. What would you think if you read that? Anybody identify an error? Eleni.
Maybe what kind of pepper seeds?
Yep, we want to be specific with kind of seeds. What else? To me, this looks like if I read that, I would think, okay, I'm going to put some seeds in a bag. Then I'm going to put 5 milliliters of hydrogen peroxide in the bag. Then I'm going to put 250 milliliters of water in the bag. So basically, I got a bag full of water with some seeds floating in it. That's what that looks like. But that's not what we're doing. So we want it to reflect what we're doing. So we would say if seeds are placed into a bag and moistened with a solution of 5 milliliters hydrogen peroxide and 250 milliliters of water. Okay, one more. If there are higher concentrations of hydrogen peroxide present. Once again, that's not telling me what I'm going to do. That's not giving me the procedure that I'm going to follow or the idea of what I'm going to do. So a better one is going to be this one at the top. If seeds are moistened with this solution right there. All right, now let's go to the then. Then the seeds will germinate faster. Well, first of all, remember that your then has to be something measurable. Is this measurable? It is. We can compare it to the other seeds, but we're not saying that here. Will germinate faster than what? Faster than seeds that are kept in the dark? Faster than seeds that are, given water? Faster than what? So I want to be more specific about that. The second one then it is the best growing solution for seed germination. Is that measurable? No, can't really measure best. What does that even mean? Is best, do I mean by that it takes the longest or it takes the least amount of time? Germinates faster or germinates last? See, best doesn't really mean anything. So I want to be more specific. Then the seeds will grow faster. That's measurable, but we're not testing growth. We're testing germination. So I would say then the seeds will germinate before the seeds in the other solutions. All right. Now, it's important to remember, You don't have to write a hypothesis in if-then format. There's many different ways to write a hypothesis. You don't have to use if-then, but it's almost always best to. In fact, some sources will say you should use this three-part hypothesis framework. This is often called the if-then because, or if-then since, or if-then due to kind of a framework. But if something is done, then something will happen because of this particular thing, the things that we would want our reader to know. Just keep in mind, however you write your hypothesis, it should explain what you expect to happen, and it should contain that independent and dependent variable. The if-then gives you that. It should be clear, testable, and measurable. All right, so let's say that we use this as our hypothesis. I will say that when you guys did your hypotheses, not very many of you used if-then format. It's really best if you can, and you can in this one. So when you can, do. So this is just a possible hypothesis. If seeds are placed into a bag with a solution of, and moistened with a solution of 30 milliliters of peroxide, 250 milliliters of water, then the seeds will germinate faster than other tested concentrations. All right, now the next thing we do is our supplies list and our procedure. This was another error that I saw. A number of you didn't include every single supply. If you forgot to put down paper towels, could someone do the experiment the way you did it? No, because they wouldn't be able to lay those seeds on the paper towel. That wouldn't be in their materials list. You have to put every single thing that you did in the procedure. And remember, you have to write what you did, not what the instructions say you could do. You can't just copy and paste what I write down as the steps that you're supposed to take. That's still plagiarizing. Plagiarizing your teacher is still plagiarizing. Plagiarizing is when you take someone else's work and try to make it look like it's your own. Not okay to do. So make sure that it is what you did in the lab, and then number your steps in your procedure. Don't repeat your materials list in your procedure. And then the best thing you can do with the lab report is hand it to somebody else. Could they reproduce the experiment exactly the way you did without you even being there? If they can't, that lets you know that you need some work. All right, the next thing in a lab report then are our results. So results is just another way to write down what you observed when you were watching the experiment. So you guys gave me your observations. There were a wide range of answers for what you observed. Some of you gave me a lot of detail. Some of you only gave me a sentence or two, which is fine if it's complete enough that I get the idea. Not many of you gave me a chart though, and charts are a great way to show your information. I recommend using a chart or graph whenever you can. So I did this experiment myself. I took some pictures of my results. So I want to evaluate them and then fill in the results sheet together. All right, so this was day 3. I didn't see any change on day 1 and 2. Day 1, I put them in the bag. Day 2, there was no change at all. This was day 3. So evaluate this. What do you see? What would you say you're observing here? Shmuel.
So far, low is the best, and high is probably second, and the medium and control are the same.
Yeah, if you look a little bit, you see a little bit of germination here in the high. There really wasn't any germination in the low. I know it's a little bit hard to tell because I've got some reflection here in the light. But yeah, definitely the low. We could say that the low germinated first. Definitely, definitely. So I would write here on day 1, no germination. Day 2, no germination. Day 3, this had germination and some growth in the low. The medium and the high had a little bit of germination in them. So that's all I need to write for my results. Now, because all we were testing was germination, we could have stopped right here. That's the end of the experiment. Once germination occurs, that's the end. But I wanted to run it a little bit longer just for fun, just to see. So I did this for another day, another couple of days. So look at day 4 and compare those results again. Now, once again, I see the most growth. They're in a different order now. So the control, there's a tiny little bit of germination, little bit of growth in the high and the medium, but a lot more growth in the low. So I measured them. I took a little ruler and I actually measured them, and I could see the low had 2.5 centimeters of growth, more than the medium or the high. That's what I put down there. Then the next day, this is my low right here. So I put down more growth and root branching, small amount of growth. You see how simple this is. I'm not putting a lot of detail into this. I'm just giving what I observed. I'm not talking about it. I'm just sharing observations. All right, so day 6 and day 7 showed the same pattern. They just continued growing. So I stopped the experiment at that point after day 7. More growth and significant root branching was happening. All right, now I looked at the whole class. I thought, let's see what our results were with the whole class. All right, so read this right here. The seeds in the control bag germinated first for 4 of you. The seeds in the low concentration germinated first for 6 of you. The seeds in the medium germinated first for 3. Seeds in the high for 2. Seeds did not germinate for 1 of you. All right, that's a paragraph. It's okay, but look how much easier it is to figure out your results when you put it in a table like this. All right, now if I use a little graph or a little graph like that, that's even clearer. I can tell just at one quick glance that the low concentration germinated first for 38% of the respondents. All right, now the thing is, there's not enough data here for us to come to a conclusion. We don't have a lot of people that are involved in this lab. This was just the 2 biology sections or honors biology sections put together. So we had a lot of differing results. We had some of them for every single one of them. And one of you didn't have any germinate at all. Why do you think we had such a wide variety of results? Aliza.
Because each one had like a few different variables than others.
Yeah. Some kinds of seeds were going to germinate faster than other seeds. Some of you would have used peroxide that was a different concentration than others. Some of you might have had different temperatures in your rooms. So all of these variables could have been factors at play here. All right, so let's talk about what we can conclude from our class results, because that's the last step in the lab report, is writing a conclusion. Remember what our hypothesis was. Now, since there wasn't a significant difference between the highest concentration, which was here, and the control, which is here, it doesn't appear as though the peroxide improved germination in the seeds, right? There's our conclusion. Since there was not faster germination using the highest concentration of peroxide, the 30 milliliter, I should say peroxide, the 30 milliliter concentration does not appear to improve germination in seeds. Okay, now in between those two, in between results and conclusion, you have your discussion. That's where you talk things out. You talk about the fact that you had different variables that were changed. You talk about the fact that maybe you used the wrong kind of seed, or maybe your bag dried out. Maybe you didn't close it up well enough. That's where you really troubleshoot and talk about why you got the results that you did. Now, this shows the results for my last 4 biology classes, 2 this year, 2 last year. So how is it beneficial to have a bigger sample size? Well, it's almost always beneficial, right? Go ahead, Shmuel.
Because we can see that this class wasn't like a freak where most people got low, like most classes got low.
Yeah.
So,
yep. So we know that we're not the anomaly here. We're in the same kind of pattern that everybody else follows. So this shows the need for duplicating lab experiments. When we do the same lab repeatedly and we get the same results repeatedly, our conclusion is more likely to be valid. Now, one of the things that I'll point out here is the media is really good at grabbing results of lab tests or studies that have been done and putting them up and saying, oh, we know now that tomatoes are bad for you. Well, how many people did tomatoes turn out to be bad for? Maybe 4? If it's a sample size of 4, you're not going to have as much validity as if it's a sample size of 4,000, right? So always look at the sample size when you're evaluating that. Tomatoes are not bad for you, but there was a time that the media was saying that they were. Tomatoes were, 4 people with past allergies. Yeah, absolutely. Very possible here. All right, so this appears to say that a low concentration of hydrogen peroxide improves germination in seeds. All right, now to review, this is what we need in our lab report. Like I said, you just did all this. You did your title, you did your purpose, you did your hypothesis, your materials list, your procedure list, results and results only, discussion and conclusion. Note that a lot of times purpose will be called abstract. It all means the same thing. Now, when it comes time to write the next lab report, use the rubric, and on your homepage, you'll see this classroom resources icon. Click on that, and right here, lab report instructions will give you a step-by-step guide to help you write your lab report. Now, I'm going to be a little stricter the next time. So just because you got an A this time doesn't mean you're home free, right? Still work to make improvements, because almost every point that was lost in the Jello lab report was because somebody didn't follow the rubric. All right. Any questions about lab reports? All right. Very good. Let's talk about cells. That's what we're studying now. So in one of the lessons last week, you guys watched a video, and I told you that we would talk about it later. So we're going to talk about it right now. You're going to see some screenshots that I took of different sections of the video, and I'll talk through them as we're looking at these screenshots. Then we'll watch the video together. All right, let's look at this now. So the video started with the scene of the blood vessel. We're inside the blood vessel here. These are the red blood cells. These are white blood cells. And this right here rolling along was a white blood cell called a lymphocyte. Now you're going to learn about all these blood cells and lymphocytes later when we study human anatomy. For now, though, just know that these are part of your immune system. They are a cell that is looking for the immune system. What do you mean? Which one is it? All right. This is a view right here of what's called the extracellular matrix. So this is on the outside of the cell. This is one cell here. This is another one right there. And there's this space in between them. And you can see how two cells that are next to each other are attached to one another. They're attached with these strong filaments that hold the cells together, keep the cells from flying out away from one another. All right, now we're going to go below the matrix and look at the plasma membrane on the top of the cell. So this is that phospholipid membrane. The phosphates are on the outside, the lipids are on the inside. These green balls on the top would be called the heads of the molecule, and then the tails that hang on the inside are the lipids. They're the tail of the molecule. There's also these rafts that float along. This is a lipid raft holding a protein that is going to reach up and make a connection with something. So they're a marker, some kind of a protein on the outside that is a receptor for something else. All right, then we move inside the cell. So with this view, we're going to go inside. We're going to see the phospholipid membrane up above us. We're going to see all these cell structures down inside. So remember, you learned about the cytoskeleton. It's made of microfilaments and microtubules. We're going to see these structural proteins that form that cytoskeleton and that the cell uses to transport things. We're going to watch them assembling and then disassembling. These are the tiny ones, the microfilaments. We're also going to see microtubules assembling and then disassembling. As soon as the cell doesn't need them anymore in that area, they disassemble and go somewhere else where the cell needs them. This is my favorite part of the whole movie. This is a protein called kinesin, and we'll watch it walk along this microtubule. And that's actually the way the molecule moves. The molecule itself looks like this, and it looks like it's walking in real life. Now it's carrying this vesicle. It contains something, some product that was made by the cell, and it's going to be taken to where it is released, to where it's needed, either inside the cell or maybe to be released to the outside of the cell. Then we look here, this is the nucleus. We're still inside the cell. This is the nucleus. These are the nuclear pores. There's going to be these little strings that come out of these nuclear pores. They are copies of the DNA, which is deoxyribonucleic acid. That's what carries the genetic information to make protein. The copy is called messenger RNA, and it comes out of the nucleus and goes to one of these ribosomes. Remember, a ribosome is a protein-making factory. So it takes little amino acids and puts them into a chain, It attaches them and the chain will come curling out this way. Note the way it curls after it's made. So after that, then you're going to see a ribosome on the endoplasmic reticulum also making a protein, because their ribosomes are everywhere in the cell. There's hundreds of ribosomes in every cell because protein making is such a big thing. Then we have the Golgi body. So you'll see the vesicles pinching off from the Golgi body after they've packaged up and processed that protein to be exported from the cell. Then they're going to be released. We'll go to the outside and exocytosis happens. That's the process, one of the processes that the cell uses to export things, to get things out of the cell. And then once again, it ends with us on the outside of the cell. We're back in the blood vessel where the lymphocyte is rolling along before it squeezes between two cells on its hunt for anything foreign that it might need. All right, let's watch this video again. I love this video. Even the music's awesome. Receptors. Now we're inside. Cytoskeleton. An enzyme comes along and breaks it apart. Mitochondria on the way. Like cartoon money. I just want you all to stop for just a second and marvel at life. Every living organism, every eukaryotic cell, this is happening inside all the time, 24 hours a day, for as many years as the organism lives. This whole process is happening and we're not even aware of it. It's just a remarkable thing. All right, so knowing these organelles is important. So we're going to go over them again. So I've given you this one. We didn't really talk about peroxisomes. That's these little yellow ones. Peroxisomes have enzymes in them that they oxidize molecules. That means they add oxygen to them. We'll talk about those later in the year. All right, so the next one is going to be this one up here. This is the energy producer of the cell. What is the organelle that is the energy producer? Shmuel.
Mitochondria.
It is the mitochondria. Think mighty mitochondrion. All right, next we have this. This is a plant cell. I can tell because it's got this big cell wall around it. It's also got these green stacked structures in there, which we'll learn all about when we learn about plants, but I can identify a plant cell because it has them. Another thing a plant cell has is a very giant one of these. Animal cells have a bunch of little ones. A plant cell has a big one. What is this organelle? Come on, guys, help him out. Can't make Shmuel do all the work. All right, Shmuel, go ahead.
Is it a vacuole?
Yeah, that's a vacuole. So the central vacuole is what we call it in a plant cell. And this is the reason plants wilt when they don't get enough water. When that vacuole loses water, the whole plant kind of gets wilty. As soon as it fills back up again, it makes the plant stand back up again. Pretty cool. All right, the next one then is going to be this space in here. The kind of jelly-like liquid and all the organelles all together. Cytoplasm, Lillian, good. So keep in mind, the cytoplasm is the cytosol plus the organelles. Just the cytosol itself, the like jelly-like substance, that's called cytosol. So this is the cytoplasm, this whole thing. All right, what's next now? What do we call these organelles that are, you guys are ahead of me. Good, Lillian. Chloroplasts. Those are the organelles inside a plant that perform photosynthesis. And they're green because they reflect green light. They have a green pigment in them. And that pigment absorbs light from the sun and converts it into a sugar called glucose. All right, moving up to the top here. These tiny little dots that are everywhere in the cell and on the rough endoplasmic reticulum. What are they called? Good, Marianne. They're called ribosomes. Ribosomes, the little protein factories. All right, next we have the place where we keep our DNA, the structure that holds DNA. Shmuel.
The nucleus, but also I had a question about the vacuole. Why does the whole plant wilt if the vacuole doesn't have enough water? Isn't there a plant cell to keep wall to keep it stiff?
There is. That's why they don't just kind of like go to nothing. If I took all the water out of you, you kind of like go down. And I mean, your bones would hold you, but they don't have bones. But yeah, it does help to keep them stiff, but it only works up to a certain point. It's still when the plant doesn't have enough water, it's still going to have a little bit of that wilting. And eventually it's going to die. And the cell walls are going to die as well. They're going to rupture. Yeah, very good question. So yes, this is the nucleus. The nucleus holds the deoxyribonucleic acid, DNA, another molecule it's important for you to remember. Inside the nucleus, we have this structure that makes ribosomes. Yep, very good, Marianne. It's called the nucleolus. You can sometimes pronounce nucleolus. I say nucleolus. Either one is correct. All right, what is the structure that these ribosomes are embedded on? Rough ER. Good, good. And then the one that doesn't have the ribosomes is smooth ER. Yeah, they have different functions as well. All right, then we have our next one, this stack of pancakes. Yep, Golgi apparatus, also called Golgi body, also called Golgi complex, whatever you want to call it. And then we finish up with the cell wall on the outside of the plant cell. Now remember, all cells have a membrane, but only plant cells and bacterial cells have a cell wall. Animal cells do not have a cell wall. Fungi have a cell wall too. All right, anybody have any questions about that? All right, I want to talk about the cell membrane. Your book really only very briefly talks about it. I think it's so important that I want to spend a little bit more time. Do plant cells have lysosomes? Yes. Yep, they would have lysosomes to break them down. Yeah, I don't have that on there, do I? But yeah, they do. All right, so just like you have windows and doors to your home that let some things in but not other things, plants have this membrane that is semi-permeable. It is selectively permeable. It lets some things out and it keeps other things in and other things from getting out. Keep in mind, every cell has a cell membrane, even bacteria. So here we have a cell right here. If we were to take a piece of this membrane right here, right from that square right there, and look at it from the side, this is what it would look like. It's a little bit of an abstract concept, but you get the idea. So pointing to the outside and pointing to the inside of the cell, we have these heads, and then in the middle are the tails. So this is a double layer membrane, and we call it the fluid mosaic model because of the way these all move around. It's like the best analogy that I have is throwing a bunch of plastic balls on top of a swimming pool. They would all change places. They would all kind of stay together, but they'd be trading places with one another. Then if you throw a bunch of beach balls in, they would represent the proteins and other molecules that are embedded in that cell membrane. They can move around too. So this is fluid. It's very flexible, but it's also like a mosaic. So we call it fluid mosaic model. Now, mostly it is composed of these phospholipids, those molecules that have the head and the tail. Those are phospholipids. They are the basic building blocks of the cell membrane. Now, keep in mind, as we're talking about all this, that we've got all these different cells, different cell types. You, as a human being, are composed of about 37 trillion cells. They all have about the same structure. Now, they all look a little different. You know, look, liver cells look a lot different than brain cells, and they look a lot different than muscle cells, but they all have the same organelles inside them. Some might have more of one kind of organelle, like obviously a muscle cell is going to have a lot more mitochondria than a bone cell. A muscle cell needs to power your movement. So it has to have a lot of energy. So it has to have more mitochondria to make that energy than a bone cell does. But they all have approximately the same structure. Now, if we take a section of that membrane from a cell and we look at it, it would look something like this right here. Now, this is the outside of the cell. This is going to be the inside of the cell. These are the proteins that are embedded in the membrane. And this is what the molecule itself looks like. If we zoom in even a little more on that molecule, this is kind of a chemical way to represent that molecule. Now, you can see that these are the ones that make up the bulk of the membrane. And you can also see why it is called a phospholipid molecule. This is a phosphate group right here, the P and the O. And these are lipids. So this part right here likes water. It's hydrophilic. These lipids, just like fats, don't like water. It's hard to mix salad dressing, right? Because the oil and water separate from one another. In the same way, the tails are going to be hydrophobic. They don't want to mix with water. Now, what happens with these is they have kind of slight charges in them. You can see those charges here. There's a positive end, and then there's a negative part right there. So that makes the slightly negative and the slightly positive part of this molecule attract water. It's polar. Remember, I think we talked about what polar molecules are. I can't remember. Polar molecules are ones that have a positive and a negative end. So this has a positive here and a negative here. So this part here wants to attract water. All right, Lillian, if we were to take a piece of the cell membrane with the cytoplasm, it depends on how big the membrane was. Actually, the membrane can sort of break open and reform again. And that happens all the time. So if you took a chunk out, yes, then the cell's going to die. All the pieces are going to go. And but if it's just a small section, if it's just a crack in it, it's going to reform itself. Yeah, good question. All right, now, some things can just go right on through that membrane. Doesn't the complementary system rip open pathogens? I don't understand the question. Complementary system. You mean complement? Is that what you're referring to?
I might be getting the name wrong. Isn't there like some part of the immune system that rips open pathogens by like poking holes in them?
Yeah. Yep. That part of that is called complement. Yeah. So I wasn't sure if that's what you were referring to. Yeah. Yep. That's one of the ways. Our bodies have living things have many ways to get rid of pathogens. One of the ways is by simply rupturing them, breaking them open. Yep. Very good. All right. So some things can just go right on through. You know, you drop them in, they go right on through. Small, uncharged molecules like oxygen. Oxygen doesn't need any help. It can go right through the membrane. Carbon dioxide can go right through the membrane. They pass right on through. Sometimes the things are bigger and they need help. So we have these proteins that are inside the membrane that are really just like a tunnel through a mountain. They're just a bigger hole so that you can get your car through. So a tunnel through the membrane just lets bigger things pass right on through. Like sodium for nerves. Yeah. The sodium potassium. We might talk about the sodium potassium pump if we have time. I think we have time for that. I'll talk about that in a second. That's a good example. Sometimes there are big things that need to pass through. Sometimes there are things that have a charge. So they might have enough of a charge, enough of a positive or negative electrical charge that they need help getting through. They do that through these channel proteins. They look kind of like this right here. So the cell membrane proteins do a lot of things. They let things pass through. They actually can send messages to other cells. And some of them change shape. You can see that happening here. They change shape in order to move substances back and forth. Now, of those channel proteins, there's 3 different kinds. There are carrier proteins, receptor proteins, and cell recognition proteins. So the carrier proteins are the ones that are going to move things back and forth across the membrane. You've got receptor ones on the outside. They're ones that send messages to other cells that are nearby. Then you've got these identification markers. These are cell recognition proteins. They're called glycoproteins or glycolipids, and they're the reason that your body knows which cells belong to you and which cells are foreign, like bacteria. So all of these markers are the same from one cell to another in your body, but they're different than any other human, unless you're an identical twin. And even then, they might be the same, or they might be different from one another. So this is the marker that identifies you. So when a bacteria comes in, your immune system goes around and looks and it says, oh, that doesn't have Lillian's marker. That doesn't have Marianne's marker. So I know I need to attack that and kill it. That's how it knows to do that. All right, let's talk about this sodium potassium pump because it's so important. This is one of the most important proteins that's in that cell membrane. It's called the sodium potassium pump. It moves sodium out and it brings potassium into cells. And it's really important because that concentration of sodium and potassium is vital for a lot of the different functions of the cell. In most cells, somewhere between 1/4 and 1/2 of all the energy that's made by the mitochondria in that cell is used to power this pump. I'm going to say that again. Somewhere between 1/4 and 1/2 of all the ATP made powers this pump. That gives you an idea of how important this pump is. Now, both of these are going to be moving against the concentration gradient. So it'd be like you trying to push a person into a room that's already full of people. That's against the concentration gradient. It's already really crowded in there, but we got to push you in anyway. It's already really crowded inside the cell, but we got to push this potassium into the cell. It's already crowded with sodium outside, but we got to get more sodium out there. So it needs energy, and that energy is in the form of ATP. Now, ATP is broken down to power the pump. After one phosphate, adenosine triphosphate, after one of the phosphates is broken off, now it's adenosine diphosphate. So that's what the adenosine triphosphate molecule looks like. This is another important molecule to remember. This provides almost all the energy needed by all living things on planet Earth. And you can watch that in this video right here. You're going to see ATP come in from the bottom and break down into ADP, leaving behind the energy needed to power this pump. So potassium is blue in this picture, the K right there, and sodium is red in this picture. It doesn't look like it in the picture, but they're moving against the concentration gradient. So let's watch and see how this happens. Note that for every 2 potassiums that are pumped out you're going to have 3 sodiums that are going to be pumped. So there go the sodiums right there. You can see your phospholipid membrane there. And this receptor, this protein channel, there's the ATP. Now it's ADP, and the pump is powered, and it moves, and it releases those. It's a pretty cool pump. And yeah, if you mess with the sodium potassium pump, if it's damaged in any way, the cell will not be able to function, not be able to do what it needs to do. All right. Sodium and potassium are not the only things that need to move across the membrane, though. There are a lot of things. We've got to get rid of the waste products. We've got to bring in nutrients. We've got to bring in the building blocks for the things that we need to build inside the cell. And cells have to send messages to one another. They have to communicate with one another in some way. And there's two main ways that they do that they let things out and let things in. One does not require energy. That's called passive transport. You're passive when you're just sitting down watching TV. You're not using any energy. Active transport does require energy. So you can think of active transport as being like pushing something uphill. Passive is just letting it roll by itself all the way downhill. Passive transmits materials without using energy. It moves with the concentration gradient. So from where it's real crowded to where it's not very crowded at all, that's pretty easy to do. Passive transport works in both directions, carries things in, carries things out. And there's three kinds: diffusion, facilitated diffusion, and osmosis. So you guys know what diffusion is. I know we've talked about that. Osmosis is passive transport. Yep, it is passive transport. It's just osmosis is water is so important in a cell, and the movement of water is so important to a cell that we give it its own special name. The diffusion of water across the membrane is called osmosis. Just gives you an idea of the importance of water. Facilitated diffusion is the one a lot of people get confused about. So make sure you understand facilitated doesn't mean helped with energy. It just means there's some kind of channel that will allow things to pass back and forth through. It's facilitated by this tunnel through the mountain, through the cell membrane, but it doesn't take any energy to do that. So it is still called passive transport. Is it like osmosis for other molecules? I guess. Yeah, I'm trying to think if that would be a good analogy. Yeah, that's okay. All right, so if something is going to move against the concentration gradient, or if it's charged, it's going to need energy in order to be able to move across the membrane. So that takes ATP. That takes that adenosine triphosphate in order to move things back and forth that are either too big or going against the concentration gradient, or maybe they're charged, but not able to go through with passive transport. So there's two different kinds of active transport. There's basically little and big. There's little ones would be the molecular transport. This would be for smaller things. And this would be the sodium potassium pump would be a good example of that. Bulk transport is when we're moving larger things through. And that uses endo and exocytosis. So we're about out of time here. Let me see here. Remember that endo means in and exo means out. They're the same process, just different directions. Exo is exiting the cell. It's something leaving. So something is made by the cell, but it's needed somewhere else in the body. Think like a hormone. You know, if you're making growth, if you're the cell that makes growth hormone, you need to get that growth hormone to all the cells in the body. So you make it, and then you have to release it. Well, remember that all those organelles in the cell, they have the exact same kind of membrane that the cell does. So when the vesicle comes along, it's got the same kind of membrane. It just fuses and it becomes part of the membrane. It releases its contents. It becomes part of the membrane. And then something else is engulfed and brought in. And the same thing happens only in reverse. Got the same exact thing happening. All right, we're going to stop there. Anybody have any questions? Cell membranes are really fascinating. Again, you know, it's just the way the cell decides what goes in and what gets out is pretty amazing. All right. I hope you guys have a wonderful week. Thank you for coming. I'll see you next time. Bye, everybody.
Bye.